Hello students, I am Dr. Divya. So in this chapter, sexual reproduction for flowering plants, we are learning about the reproduction in the flowering plants. Especially we are learning about sexual reproduction in flowering plants. So in the last class, we have learned some concepts of this chapter. So what is reproduction? It's the biological process by which new individuals or new organisms are produced from their parents. Okay, so new organisms are producing from their parents. That process that is called as reproduction. So that reproduction means formation of new individuals or new organism from their parents. So why reproduction should take place? Or what is the purpose of this reproduction? For the continuity of species, right? For the species continuity, reproduction is taking place. So mainly for species continuity from generation to next generation. So generation after generation, for the continuity of a particular generation or a particular species through generations, that is called as that process for that continuity, that is called as reproduction. So there are two types of reproduction mainly, sexual reproduction and asexual reproduction. So what is sexual reproduction? In this chapter we are going to learn about the sexual reproduction, right? So already in previous classes we were learning about few terms and few processes of this sexual reproduction. So what does sexual reproduction mainly? Sexual reproduction involves two parents. That is the important point. Sexual reproduction is the involvement of two parents. There is an involvement of two parents, and these both parents, two parents are involved, right? So both parents are sexually opposite. So in the sexual reproduction, the production of male gamete and female gamete will be there, right? Male gamete and female gamete should form in sexual reproduction. So this is called as gametogenesis. So first step of sexual reproduction is the formation of male and female gamete. This is called as gametogenesis. Genesis of gametes, which are all the gametes, male and female gametes. So these gametes are formed. This process is called as gametogenesis, formation of gametes. Then next step, first step is the formation of gametes or the gametogenesis. Next step, already gametes are formed. These gametes should fuse. Then fusion of both male and female gamete. That is the second step. Okay, this is called as fertilization. First gametes will form. Next fertilization. Fertilization means these formed male and female gamete will fuse together. That process is called as fertilization. So such stored, these male and female gametes that formed male and female gametes will fuse together, and this fertilization, that process which is called as fertilization, it is having several steps. Okay, that fertilization process include pre-fertilization, then fertilization and post-fertilization. This is not a single step, this fertilization. That is involving complex processes: pre-fertilization, fertilization and post-fertilization. I said in sexual reproduction of flowering plants, three major events are there, right? Pre-fertilization, fertilization and post-fertilization. So in pre-fertilization, what and all included, or what and all we learned under the title of pre-fertilization, or what we are going to learn under this pre-fertilization title. So in this we are going to learn about structure of a flower, then male reproductive structure, male gametogenesis, female gametogenesis, then embryo sac formation, pollination, then pollen pistil interaction. So all these topics are coming under pre-fertilization events. What do you mean by pre-fertilization event? Before fertilization, what and all events are taking place? Pre-fertilization, before fertilization. So a plant or a flower, it is getting ready for that fertilization, right? So before fertilization, what and all processes are going on, all those are coming under pre-fertilization events. Okay, so in this pre-fertilization we are learning about flower structure, male reproductive structure, then male gametogenesis, female reproductive structure and female gametogenesis, then embryo sac formation. Okay, then formation, pollination, then pollen and pistil interaction. So all these things are coming under pre-fertilization events. Then next is fertilization. So under fertilization we will learn about the process of double fertilization and zygote formation. Fertilization means fusion of this male and female gametes. So during pre-fertilization we are learning about the process of male and female gamete formation. Then when it is coming to fertilization, already these formed gametes will fuse together and that will lead to the formation of zygote, right? Then post fertilization, that includes, already zygote is formed, the zygote will develop into embryo. So in post fertilization we will learn about embryo formation, then seed formation, that embryo, then development of seed, then fruit. Okay, so all these are coming under post fertilization events. Post fertilization means after fertilization. So first we learned about flower, right? So flower is having so many usages, we know that, like for decoration, then for perfume, all these we are using flowers. Then flowers, they don't have only one function, or it is not only for one purpose. So flower, it is a for multiple purposes we are using that flower. So flower is a modified shoot. Okay, modified shoot, and it is a site of sexual reproduction, and that is the reproductive structure. Flower is the reproductive structure of angiosperms. Then we have learned about parts of a flower, right? Pedicel, thalamus, just remember the structure of a flower. Pedicel, then thalamus, then ovary, then sepal, petal, stamen, style and stigma. So that's the structure of a flower. Then we learned about all these. Then sepals are collectively called as calyx. Petals are collectively called as corolla. Then carpel is the female reproductive part of a flower. Female reproductive part of a flower, that is the carpel. And this carpel consists of ovary, style and stigma. Ovary, the bulged portion. Then style and the top portion, stigma. So carpel consists of ovary, style and stigma. Then inside this ovary, ovules will be present, right? So all these things we explained in the previous class. So the female reproductive part, carpel, it consists of ovary, style and stigma, right? Here you can see in this flower, ovary, style and stigma. Then inside ovary, ovules will be present. Here you can see ovule also. Then gynoecium. Gynoecium is the female reproductive part. Okay, female part of the flower. That will be having one or more pistils. Okay. Then we discussed about androecium, the male reproductive part, or that is the collection of stamens. All stamens together, they are called as androecium. This calyx and corolla in a flower, they are called as non-essential whorls. Androecium and gynoecium are called as essential whorls. Why? Without androecium and gynoecium, because androecium is producing the male gamete and gynoecium is producing the female gamete. So without androecium and gynoecium, reproduction is impossible. It's not possible. Okay. So we learned about essential and non-essential whorls. Then structure of stamen. We learned about structure of stamen, which consists of anther and filament. Stamen consists of anther and filament, and this anther, then we concentrated into the portion of anther which is bilobed, then structure of microsporangia, which is having epidermis, endothelium, middle layers as the layers. So the layers of anther, that also we have learnt, epidermis, endothelium and middle layers. Then we discussed about pollen mother cell and the formation of microspore from pollen mother cell. That is called as microsporogenesis. Pollen mother cell formation, then from that microsporogenesis also we learned. Then we discussed about the development of pollen grain and structure of pollen grain also we learned. Pollen grain is having two layers, outer exine and inner intine, and how it will mature, and at maturation what will happen, all these things we discussed. So at maturation, that pollen grain will be having two cells, the vegetative and the generative cells. In that, vegetative cell is bigger and generative cell floats in the cytoplasm of vegetative cell. So all these things we discussed. Then we learned about pollen allergy. Some pollen grains will be allergic to some people. Then pollen grain as a source of nutrient, that also we learned. For athletes and all, they are using in tablet form. So up to that we discussed in the last class. Now the androecium part got over. The male reproductive part, androecium part got over. Next, the male gamete part over. We have learned about the pollen grain. So next is the female part. Female part, the gynoecium. Okay. Next we are going to learn about the female part, the gynoecium. And gynoecium is the female reproductive part. Okay. Gynoecium that may have single pistil. So it is called as, or gynoecium is otherwise called as carpel. Okay, the carpel, so many carpels, they are collectively called as gynoecium. It may be having one or many carpels. So together it is called as gynoecium, the female reproductive part. So when it is having only one carpel, it is called as monocarpellary, or more than one means it is called as multicarpellary. Here you can see monocarpellary as well as multicarpellary. One means it is called as monocarpellary, or gynoecium sometimes they may have more than one pistil or more than one carpel. It is called as multicarpellary. So if there are more than one pistil, sometimes that may be fused. That condition is called as syncarpus. Okay, that will be fused. That is called as syncarpus condition. And in some cases, that pistils may be free. It is called as apocarpus. Okay, sometimes it will be fused, called syncarpus. Here you can see syncarpus, and sometimes it will be free. That means apocarpus. Okay. So here in this picture, one is a dissected flower. Here this is a dissected flower. You can see this is hibiscus. In the structure of dissected flower hibiscus, here you can see the thalamus, then the ovary, then this is a style. Here you can see a long style, and here stigma, free stigma you can see. So thalamus, this bulged portion is ovary, then this long style, and here stigma. Okay. Then in the second picture, it is papaver, poppy plant, this one. Here you can see stigma. Here it's in a fused form. And third one is Mechelia. Mechelia is here also you can see the carpels. Right. So the gynoecium represents the female reproductive part of the flower. And the gynoecium that may consist of single pistil, it is called as monocarpellary. And if gynoecium is having more than one pistil, if it is having only one pistil, it is called as monocarpellary. Mono, that word indicates single. So monocarpellary, it is having only one carpel. And if the gynoecium is having more than one pistil, that is called as multicarpellary. And in multicarpellary condition also there are two types. Multicarpellary, if it is having more than one carpel, in there also different types are there. So when there are more than one pistil and it is fused together, it is called as syncarpus. Okay. Or if they are free, it is called as apocarpus. Multicarpels are there. If it is fused, it is called as syncarpus. And in multicarpellary condition, if it is free, that is called as apocarpus condition. Okay. Then next is the structure of pistil. Okay, structure of pistil. Here each pistil has three parts. Okay, in the diagram you can see each pistil has three parts. See, in this structure, in the floral parts, you can see the floral parts. Here this is the pedicel. Then above that, here you can see the ovary, and inside ovary you can see ovules, and these are the sepals, this greenish color sepals, and this colorful part, this is the petal. Sepals together they are called as calyx, and petal. Petals are collectively called as corolla. Then here you can see the stamen, this one. Then here style, this long part, this is a style, and this is stigma. Stigma, here you can see stigma. On stigma the pollen grains will come and sit. So that is the position where pollen grains will land, on stigma. So from that we are zooming this portion, the center portion, the stigma, style and ovary. So when we are discussing about stigma, style and ovary, look at this picture. Each pistil, it is having three parts: stigma, style and ovary. This is the stigma, style and ovary. Okay, the stigma, that is the place where a pollen grain lands. On stigmatic surface the pollen grain will land, right? Next is style. So the elongated part, elongated and slender part below stigma, it is called as style. So below stigma style is present. In all flowers it won't be so much elongated. In hibiscus you can see an elongated long style. Normally it will be longer, but in all flowers it won't be having so much length. So here you can see the style. This is the elongated and slender part, and it is below stigma. Top portion is stigma, then comes style. Style is the elongated slender part. Okay, and this style is the one which is connecting the stigma with the ovary. Okay, style is the connecting part between the stigma and the ovary. And next is ovary. So inside this is the bulged part. This ovary is the bulged part, and inside this ovary ovules will be present. So here style is the connecting part between stigma and ovary. So when a pollen grain is landing on the stigmatic surface, How that male gamete will reach to the ovary? Because female gamete is present inside the ovary, right? Ovules are present inside the ovary. So a male gamete from the pollen grain, it will reach to the ovary through style. So each pistil has three parts: the stigma, style and ovary. So the stigma, that serves as a landing platform for a pollen grain. Stigma serves as a landing platform for the pollen grain. And the style is the elongated slender part beneath the stigma. Style, you can see the style here. This is the elongated and slender part beneath the or under the stigma, below the stigma. Then see this here, hibiscus one portion, you can see this is the portion which I am talking about. And the basal bulged part of this pistil, that is the ovary. Here you can see the ovary, the basal bulged portion, that is the ovary. Okay, so stigma, style and ovary, these are the parts of this pistil. Above, top is stigma, then comes the style, and down the bulged portion is ovary. Style is the portion which connects the stigma and ovary. Then inside the ovary, you can see inside this ovary, ovules are present. Okay, so we are going to see inside the ovary how it will be. So I'm going to explain that now. So down the bulged part of this style, you can see the ovary, right? So inside this ovary, see this diagram, you can see down, you can see the ovary here, and in this also you can see the ovary. So inside the ovary, ovarian cavity is there. This is the ovarian cavity. Okay. So ovarian cavity is also called as locule. Ovarian cavity is also called as locule. Okay. So inside the ovarian cavity, placenta is present. Where placenta is present? Inside the ovarian cavity, placenta is present. And you have learned different types of placenta, right? Which and all the placentation, about the different placentations you have learned. Axial, parietal, basal placentation, all this you have learned in the 11th standard chapter, right? So from these placenta, ovules are arising. So ovules are connected with the placenta, and from these placenta the ovules are arising, or ovules are attached to this placenta. Okay, here you can see the ovules are attached. It is attached to the placenta. Ovules are otherwise called as Megasporangia. Another name of ovules, it is Megasporangia. They are commonly called as ovules. So ovules are otherwise called as Megasporangia. And number of ovules in an ovary, here you can see, it depends upon the plant species. Okay, number of ovules in the ovary varies. There will be difference in the number of ovules in the ovary. So in the case of rice, wheat, mango, all these things, you can see only one ovule in an ovary. Mango, you can see only one ovule. Then in the case of rice also, there is only one ovule in the ovary. Rice, wheat, mango and all, one ovule will be present in an ovary. But in papaya, watermelon, orchid, all this, you can see many ovules in an ovary, right? In the case of papaya, if you are cutting papaya, you can see many seeds, many black color seeds. So there are many, that is the locule. Inside that locule, you can see many seeds. Same case in the watermelon, you can see many seeds, black color seeds or white color seeds you can see inside the watermelon, right? And in orchid, watermelon and in papaya, many ovules are present in the ovary. So inside the ovary, it is the ovarian cavity or locule. It is otherwise called as locule. So in the placenta, it is located inside the ovarian cavity. Placenta is located inside the ovarian cavity, and arising from the placenta are the megasporangia. Megasporangia, or otherwise it is called as ovule. Ovules are arising from the placenta. So from placenta, megasporangia is arising. Megasporangia is commonly called as ovule. Ovules or megasporangia. And the number of ovule in an ovary may be 1. In the case of wheat, paddy, mango and all, number of ovule in an ovary, it is 1. So in wheat, paddy, mango and all, you can see only 1 ovule in an ovary. But in the case of papaya, watermelon, orchid and all, you can see many ovules in an ovary. Okay. So here in this picture you can see the ovary wall, then inside ovule, these ovules are attached to the placenta. Then this is style and stigma. And here parts of flower I have already told. Next we are going to learn about the structure of ovule or megasporangium. So now we are going to learn about the structure of a typical angiosperm ovule. Okay, a typical angiosperm ovule, we are going to learn the structure of the typical angiosperm ovule. So in the first diagram you can see anatropous ovule. Anatropous ovule inside a pistil. This is the pistil. Okay, see here we have told this the structure of a pistil. So inside this pistil, that part we are zooming, this is the pistil. So inside this is ovarian cavity, and inside that one ovule. Okay, this ovule is connected to the placenta. Okay, so this is the ovary part, and this is the style, and here stigma. So here pollen grain lands. So inside this ovary, this is the stigmatic surface where pollen grain lands. So here inside this ovary, ovule will be present. Right, here you can see this is the ovule. So now we are going to learn about the typical structure of anatropous ovule, that ovule which is present inside the pistil. So see the outer part. Okay, so in this diagram you can see stigma. Here you can see stigma, next is style, and down you can see ovary. Right, so inside this ovary you can see an ovule. See, this is the ovule, and this ovule is connected here to the placenta, and different types of ovules are there. So this is called as this, which ovule I am going to explain, that is anatropous ovule. Okay, so after explaining about the common structure of ovule, I will tell you about different types of ovules. Now listen. So inside ovary you can see an ovule. Right, this is ovary wall, and inside ovary we can see ovule. So this ovule, it is a very small structure. So in some diagram you can see ovule is attached to this placenta by a stalk. Here you can see. See, this is the ovule, and this ovule is attached to the placenta by a stalk. Right, and that stalk is called as funicle. This is called as funicle or funiculus. Here we have marked as funiculus, funicle or funiculus. So the ovule which is attached to the placenta by a stalk-like structure, that is called as funicle or funiculus. Important MCQ. Then the body of the ovule fuses with funicle in a particular region. See, already we have told this funicle. It is a portion where an ovule is attaching to the placenta by a stalk-like structure. This is a stalk-like structure called funicle. Right? And the body of ovule fuses with this funicle, and that fusion is in a particular region. Okay, that region is called as hilum. Okay, when here see, this is exactly upside down structure. Here this is another type of ovule. See, this is a vertical section through carpel, and this is anatropous ovule, another type of ovule which is common in angiosperms. So we are going to learn the structure of an anatropous ovule. So in this anatropous ovule, first you can see the funicle or funiculus, which is attached to the placenta by a stalk-like structure, that is called as funicle, right? And the body of that ovule, which is fusing with the funicle at a particular region, and that region is called as hilum. See, the fusion of that body of ovule with the funicle, that is in a particular region, that fusion, that region is called as hilum. You can see here, this is the hilum region, that body of ovule, it is attaching to the funicle. Here, this is the funicle, stalk-like structure. So that place of union, that is called as hilum. So this hilum represents the junction between ovule and funicle. Just now I said that joining area or that point, that is called as hilum. So hilum represents the junction between ovule and funicle. So each and every ovule has one or two protective envelopes. You can see all ovule that is having a protective envelope or protective covering, that is called as integuments. These coverings are called as integuments. Okay, here you can see these are the protective covering called integuments. So this integument covers the nucellus except at the tip. Okay, so except here you can see this integuments covers the nucellus except at the tip. The tip portion won't be covered, except that all portions it is covering these integuments. So at the tip, a small opening, you can see here a small opening at the tip, that is called as micropyle. This opening is called as micropyle. Here we have marked as micropyle. So except at the tip, everywhere this covering is there. These integuments are covered. And at the tip there is a small opening called micropyle. So that end where micropyle is present, that side is called as micropylar end. Here the small opening is there, that is called as micropyle, and where that opening is there, the micropyle is there, that end is called as micropylar end. Important, all these terms you need to remember because all these are important MCQs. So what is hilum, what is funicle, what is integument, then micropyle, what is micropyle, and what is micropylar region. So the end where micropyle is present, that is called as micropylar end or micropylar region. And opposite to micropylars, now here this is the micropylar region, and opposite to micropylar region, that is the chalaza. And where that chalaza is present, that side is called as chalazel pole. So this is the micropylar region or micropylar pole, and the opposite is chalazel region or chalazel pole. Okay. So the chalaza represents the basal part of the ovule. Chalaza represents the basal part of the ovule. So the end where chalaza is present is called as chalazel end. Where chalaza is present is called as chalazel end, and it is exactly opposite to the micropylar end. Okay, so the ovule, it is a very small structure and it is attached to the placenta by a stalk which is called as funicle. This is the funicle, the stalk-like structure, and the body of the ovule that fuses with the funicle. The body of the ovule fuses with the funicle, and that is called as hilum. Okay, that region of fusion, that is called as hilum. This hilum is representing the junction between ovule and funicle. So the junction between the ovule and funicle, it is called as hilum. Don't get confused between hilum and funicle. Hilum is the junction or that union where that point, the point of that union, that is called as hilum. But funicle is the stalk-like structure. Okay. Then each ovule that has one or two protective envelopes called integuments. So each ovule is having one or two protective layer called integuments. Here you can see outer integument and inner integument, two layers. Okay. Then integument that will leave an opening that is called as micropyle, and where the micropyle is present, that region is called as micropylar region, and exactly opposite to the micropylar region, that region is called as chalazal region or chalazal end. Okay, and next the integuments that covers a mass of cells, they are called as nucellus. Here I said integument will cover the nucellus. I said in the previous, while explaining the previous slide, I said integument will cover the nucellus. Nucellus is nothing but the mass of cells that is called as nucellus. The inside the integument, mass of cells will be present, that is called as nucellus. Here we have marked nucellus. So this portion, this is called as the surrounding portion inside, which is present inside the integuments. These are called as, this area is called as nucellus, or those cells are called as nucellus. Integument that covers a mass of cells called nucellus, and this nucellus that forms the body of ovule. Okay, so nucellus forms the body of ovule, and nucellus is the mass of parenchymatous tissues. Okay, nucellus is composed of parenchymatous cells or tissues, and this is enclosed within the integuments. Nucellus is enclosed within the integuments, and it is composed of parenchymatous cells. Okay, cells or parenchymatous tissues, and actually they are the protective envelopes. And nucellus is with abundance food material. Okay, lot of food material it is present in the nucellus or the tissues of this nucellus, and that is the main part of ovule. Okay, so this integuments, they are the protective layers, and inside in integuments, nucellus, which is a mass of cells or tissues, and this mass of cells or tissues that is with so much of nutrients. Okay, or with abundant food material, that mass, it is called as nucellus, and nucellus is the main part of ovule. And the female gametophyte or the embryo sac is located in the nucellus. Here you can see this is the embryo sac. So this embryo sac is located in the nucellus. Why embryo sac is located in the nucellus, and why nucellus is composed of nutrient cells? Because this developing embryo is getting nutrients from the nucellus. Okay, so that is having nutritive function. So an ovule generally has a single embryo sac, and which is formed from a megaspore. Okay, it is formed from a megaspore. Clear? So the integuments encircle the nucellus except at the tip, where a small opening called the micropyle is organized. And opposite to the micropylar end, there is a chalaza, and which is representing the basal part of that ovule. Got it? And next is based on the nucellus or the development of nucellus, there are two types of ovule. Okay, so based on the nucellus or the development of nucellus, based on the development of nucellus or based on the nucellus, there are two types of ovule. You can see two types of ovule. First one is quasi-nucellate ovule, and second is tenunucellate. And first one is quasi-nucellate ovule, and second one is tenunucellate. First one is quasi-nucellate, and second type is tenunucellate. So what is crassinucellate? So in this type, the nucellus is well developed. There is a well developed nucellus or the presence of massive nucellar, massive nucellus you can see. Okay. So in the nucellus, where a well developed nucellus is present, that is called as crassinucellate, or the presence of massy nucellus. Okay, there will be massy nucellus or a well developed nucellus, that is called as crassinucellate. And you can see this type, and it is composed of many layers. In this type you can see it is composed of many layers, and there is a well developed nucellus, that is called as crassinucellate. Okay. In polypetalous you can see this well developed nucellus. And second type is TeenEinstein. Okay. So in TeenEinstein type, that nucellus is it's not well developed, or it is very less developed nucellus you can see, or nucellus is single layered in this type of ovules. Okay. So in this type of ovules, the nucellus is single layered and it is present in gamopetalous. So crassinucellate condition is present in polypetalous, and TeenEinstein condition is present in gamopetalous. Note down what is crassinucellate. So depending upon the development of nucellus, how this nucellus is developed, ovules are of two types. There are two types of ovule. First one is crassinucellate ovule and second one is TeenEinstein ovule. In crassinucellate ovule, that nucellus is well developed. Okay, and it is present in polypetalous condition. In polypetalous you can see this kind of ovules. And TeenEinstein ovule means in this nucellus is poorly developed. There is no well developed nucellus in the case of TeenEinstein. And you can see this TeenEinstein ovule in gamopetalous. Okay, so here hilum you can see, then comes the funicle, micropylar region. The integuments are present. Integuments will leave one portion, that is a micropylar region, and the integuments, outer integument is there, inner integument is there. Then embryo sac you can see. This embryo sac is embedded in the nucellus. Nucellus is parenchymatous. All these are important points. Nucellus is parenchymatous and it is having nutritive function. Embryo sac is present in the nucellus because it is getting the nutrients from the nucellus. Nucellus cells have nutritive function. So when this nucellus, based on the development of this nucellus, there are two types of ovule, crassinucellate and TeenEinstein. Crassinucellate means there is a well developed nucellus in that. In polypetalous you can see that condition, and TeenEinstein means there is poorly developed or there is no well developed nucellus. This in gamopetalous condition you can see. Clear? And next is based on the integuments. Based on the integuments, ovules are classified as unitegmic ovule, bitegmic ovule and ategmic ovule. Okay. So based on the first, just now we explained based on the development of nucellus, and now this classification is based on the integuments. Unitegmic ovule, bitegmic ovule, ategmic ovule. Important NEET questions, note down. Unitegmic ovule means presence of a single integument. Unitegmic, single integument, presence of a single integument. For example, members of gamopetalae and gymnosperm. In the members of gamopetalae and gymnosperm, you can see unitegmic, that means with single integument. Next category is bitegmic ovule. That means presence of two integumented ovule. Okay, there will be two integuments. Here you can see outer and inner integument. Like that, two integuments will be present. So most of the angiosperm, that means polypetalae, capsula and monocots. In that we can see bitegmic, two integuments, presence of two integuments, that is the bitegmic condition. Next is ategmic ovule. In that there won't be any integuments. So ovule which are devoid of integuments, example Olax, Liriosna, then Loranthus, all these are example for ategmic or without integuments. So there are, based on the integuments, ovules are classified as unitegmic ovule, that means presence of a single integument. Example, members of gamopetalae and gymnosperm. Next is bitegmic ovule, that means presence of two integumented ovule. Then example, most of the angiosperms. Then third type is ategmic ovule, ovule which are devoid of integuments. There won't be any integument. The integument is absent in such cases. So Olax, Liriosma, then Loranthus, Centella. All these are the example for ategmic ovule. Clear? So based on nucellus, development of nucellus, it's classified into two, and based on the integument, ovules are classified as unitegmic, bitegmic and ategmic. So that's all about today's class. So we have gone through the female part, gynoecium of plants. Then we learned the structure, then structure of ovule. All these things we have learned. So that's all. Thank you.